to get rid of this person, we want to learn about cochlear implants. But I hope by the end of this I'm able to convince you that it's very important for us to understand physiology and anatomy as it's relevant to cochlear implants, and that's what you're doing right now. Listening to me is all about auditory physiology at some level. It's, when you look at it at the cell and the nerve level, it is all about physiology. And when we want to understand why things are not working the way we want them to work, we need to understand what's happening at the level of the neurons. Now I need not tell anybody about this, and I'm going to skip over everything that as it relates to hearing and not to cochlear implants. We all know this and we're very familiar. I'm going to gloss right over it. We also know the functions of the outer and the middle ear, very important for us, but when we think of cochlear implants, probably they're not that relevant. So I'm going to go right over that also. And this, where it starts getting important, we all know that we need to understand the fine structure of the cochlea, because that's where the cochlear implant is actually going to go. And this famous, this is also important. May I have a pointer, please? It's very important for us to remember that the cochlea is like this, literally like a spiral staircase. And right in the middle you have the modulus. And the modulus is very important because that's where all the neurons congregate and go to form the auditory nerve. And where we want to get as close to this modulus with the electrode that we put in during cochlear implant, and the electrode actually goes right round into the scala tympani, from where it has very easy access to the nerve fibers. Now, if we look at the cross section of the scala media of the cochlea, that's where all the action is taking place. These points are very important, the points that I've marked in blue over here. Here is where the tectorial membrane is starting from the spiral limbus. Here is the pillar, here is part of the basilar membrane, and this is where the pillar of one of the pillar cells is. This is the arch of Corti, and over here where normally in the normal ear a lot is happening, because that's where the tectorial membrane and the outer hair cells meet. And if you understand how all of this works, you understand what goes wrong many times in cochlear hearing loss and about cochlear implants as well. Now, if you go closer to that organ of Corti, this is very important because here is where a lot of the hair cell transduction is happening. And here is the inner hair cell, which works very differently from the outer hair cell. And what's very special about all this is, what I love always saying, that the entire cochlea that you saw before, opened out, is only 3.5 centimeters long, which is just about this much. And on that you have about 12,000 outer hair cells and about 5,000 inner hair cells. And all of this structure is moving all of the time that we're hearing. That's absolutely all the time. And usually this structure is more or less functional for all of our lives. So you can imagine how strong this structure is, though it's really very small. If you go to the surface of the top part of the hair cells, here are these little stereocilia. These are the outer hair cells and those are the inner hair cells. And this W structure and the peculiar formation of the stereocilia is very important for normal transduction. Now, one very important part of the system, which is very important when we think about multi-channel cochlear implants, is something called tonotopic organization, which is the anatomic arrangement by frequency, which is maintained right from the cochlea up to the primary auditory cortex. Then we know that the basal-most part of the cochlea is sensitive to the highest frequencies and the apical-most end of the cochlea is sensitive to the lowest frequencies. Hence, the nerve endings at the base of the cochlea are more sensitive to high frequencies, whereas those at the apex of the cochlea are sensitive to the low frequencies. And this is very important when you're putting an electrode inside and you want to get close to the correct neurons. Another very important thing about the cochlea is the potentials of the cochlea. The endolymph is very high in potassium. It has a positive charge of about 80 microvolts, which is probably the highest positive charge in any cell in the body. And inside the hair cells here, you have a minus 70 millivolts charge, so the potential difference across the surface is about plus 150 millivolts, which is the highest potential difference anywhere in the human body. And it is this potential difference that keeps the ear going. And what is responsible for this peculiarly high positive potential is the stria vascularis. Again, a very important part. And you see that whenever the stria vascularis is not functioning, like for example, there are certain autotoxic drugs that do not allow the structure to function, it does not pump back the potassium ions which are required for this process to occur. And that's when you can start getting a hearing loss. And it's another reason why they say that children with connexin 26 mutations, one of the genetic mutations responsible for hearing loss, they do very well with implants, because the only problem in that mutation seems to be that the stria vascularis is not able to do this job of recycling the potassium ions, and the rest of the structure is fine. So any kind of pathology which leaves all of the structure and the nerve endings below intact is obviously a great case for a cochlear implant, because there's nothing else wrong, no ossification, no nothing. So you know that the cochlear implant has every chance of working very well. Now a little about the travelling wave. What actually happens in the cochlea? So when you stimulate the oval window, you have a travelling wave, which always, as the travelling wave paradox says, it always travels from the base to the apex. And as it travels, it has a maxima at some point of the basilar membrane which is most sensitive to that frequency. So going back to the tonotopic organization, you see that high frequency sounds have a travelling wave maximum at the base of the cochlea, whereas the low frequencies travel right up. Now while the basilar membrane seems to have reasonably good tuning, this tuning does not explain why we are able to make out such fine frequency differences. And then we go further. This is another diagrammatic representation of the travelling wave. Now the outer hair cells are very, very peculiar. They're very unique because they are sensory, but they move. It's a sensory cell that contains muscle proteins and has a very complex lateral wall structure. The OHCs, or the outer hair cells, contract and stretch as the basilar membrane moves. So it's kind of like jumping on a trampoline. So if you're jumping on a hard surface, you don't really get so high. But if you're jumping on a trampoline, the trampoline is also moving and you're also moving and you're also bending and stretching. So obviously you go much higher. So actually they say that because of the cochlear fluids, there's so much friction that actually there shouldn't be such good transduction of the sound when the oval window is hitting against, the stapes is hitting against the oval window. It's this dynamic movement of the outer hair cells which is actually helping this process to get even more effective, and this entire system is often known as the cochlear amplifier. Though there are actually, of all the afferent nerve fibers, only about 5% of the fibers supply the outer hair cells, but yet the outer hair cells are responsible for the fine-tuning of the cochlea as well as the sensitivity to soft sounds. The outer hair cells, as I said before, contain muscle proteins and the property of electromotility. They also have most of the efferent, that is the control from the higher centers down, most of those efferent fibers are ending on the outer hair cells. So it's very important for regulating the functioning of the cells. And these outer hair cells are thought to be responsible for cochlear autoacoustic emissions. Now if you look at what happens at the level of the cells when the basilar membrane moves, the basilar membrane has a displacement force, and this, because these outer hair cells are, the outermost cilia are embedded inside the tectorial membrane, they have what is called a shearing force. And the basilar membrane is anchored quite firmly on one side, and so also the tectorial membrane. But as you go further away from this pivot point, this part is able to move much more. So as this basilar membrane moves up and down, the hair cells also move, contract and stretch, and that's what helps these little ionic channels over here to open. So the hair cells are stimulated due to this bending and shearing action. Now if you look over here, if you look very finely at the outer hair cell, these stereocilia are further connected by what are called tip links. And when this basilar membrane moves up and down, it's like the stereocilia are kind of caught between two membranes and they are moving out and in. When they move outwards, that's away from the modulus, at that time there is depolarization occurring. And when it moves towards the inside, there is hypopolarization occurring. So when there is depolarization, there is movement of the ions, and that's when a potential can be generated. The inner hair cells, on the other hand, have no contact with the tectorial movements. So they are only stimulated by the velocity of the fluids that move in the cochlea, which is why they are not very sensitive to soft sounds, whereas outer hair cells are. But of course, we need the inner hair cells too. They all both have different roles to play. Now, what are the potentials that get generated inside the cochlea because of all of this movement? The first is the cochlear microphonic, which is almost like a mimicking of the signal. So if the signal is a low frequency signal, the cochlear microphonic will look exactly like a low frequency signal. If the signal increases, the microphonic increases. There is then the summating potential, which has the outer hair cells and the inner hair cells responsible for it. So if you compare these potentials in measures such as ECOG-G, you can figure out whether the outer hair cells seem to be a problem or the inner hair cells. Then you have the action potential, which is generated by the nerve itself. The auditory nerve is containing different types of fibers. There are, as I said before, only a very small percentage of fibers which are ending on the outer hair cells and the rest end on the inner hair cells. And there is also the efferent auditory system, which is the control of the higher auditory structures on the outer hair cells and the inner hair cells. Now, every nerve fiber has what we call a tuning curve and a characteristic frequency. Why do we need to know about this here in cochlear implants? Because we want to stimulate the right neurons. So you want to get close to those neurons which are sensitive to that particular frequency which you are trying to transmit. Now, if you look here, this is the tuning curve of a neuron, and each color is representing a different neuron. So you see that here the neuron can get stimulated with very low intensity signals, but as you go away from that frequency, the response of the neuron gets poorer and poorer. So for example, this green dot is a small intensity signal at a particular frequency. So only the blue neuron is able to respond to it because it's out of the response area of the other neuron. But when you go higher in intensity, you find that over here there are two neurons which can respond. You go over here and all of the neurons are able to respond. So you see that as you increase the stimulus level, more and more neurons are coming in and acting. So as you increase the stimulus level, it's not only the rate of firing of each neuron that increases, but a larger number of neurons are participating in the entire process. And that's how loudness increases as you increase the intensity. Neurons also have this ability to lock themselves to the phase of a signal, and this property is very important for coding temporal cues of the stimulus. Like if I say pa and ba, you know that these two are different only by the voice onset time. So the timing of the firing is very important in our auditory system, and one of the places where this is happening is at the neuronal level through phase locking. Now, the dynamic range of each neuron is only about 40 dB. So by 40 dB, it saturates. But the entire system gives us a dynamic range of 120 dB or so, or more than that actually, because many neurons are working together. Now, the fact that we keep coming up with new processing strategies shows that we don't really yet understand the auditory physiology completely. Now, another thing which I think many of us who attended Dr. Hall's lecture last week heard, we hear with our brain. And how do we know that? Because we hear sounds and understand speech when electrical stimulation is provided either to the auditory nerve through a cochlear implant or to the cochlear nucleus, where the traditional hearing structures or ear is not involved at all. So all fine acoustic cues such as intensity, frequency, time, all have to be maintained at higher levels of the auditory system, most of which is neural. So you have the place principle, which talks about how the place is important in the cochlea, and the temporal theories which believe that timing is important, and we know that both of them are important. And if you know this as an audiologist, you can adjust math parameters far more effectively and logically rather than going just by trial and error. Hence, there are 2 points that I need to stress here. The cochlea and nerve are not enough for complete auditory functions, and that detection is not the only thing that you need to do with your auditory system, but there are higher auditory centers which are doing far more complicated auditory function. So what does this mean to us? It means that if there is auditory deprivation, it affects not only the cochlea and spiral ganglions, but also the higher central processes. So even things like listening in noise, which is something that happens at the lower brainstem due to binaural interaction, they say that this is affected in children even with long-standing conductive loss because these processes mature only with auditory inputs. And this might also explain why some children seem to have your NRTs and everything perfectly fine. They have fantastic CI-assisted audiograms, but are still incapable of getting good auditory speech perception. So the point is there something more that could be wrong? So we know that you can have functional abnormalities without something wrong in the actual structure as is seen on a CT or MRI. We also know that peripheral hearing loss can coexist with central pathologies. So whenever we see a child who doesn't seem to be doing very well, though he is listening, seems to be listening well with a cochlear implant at the detection and discrimination level, but is not doing higher auditory skills, then you need to really think, is something more going on with this child? Now if you go, now I'm going to talk about the auditory pathway, because though this is a simplified diagram, it's important to remember a few things. One is that after the level of the lower brainstem, there's information from both sides going on, and what is not shown here is that a lot of crisscrossing of fibers that happens, which is very important for us to do many of the complicated listening functions that we do. So I'm going to again say very briefly about different things that happen in the auditory system. One feature of the auditory system is tonotopic organization. Very important to keep frequency cues intact. Also, there's redundancy. That means if one pathway is affected, there's always an alternative pathway. What the auditory nerve does is just breaks up the signal into smaller components, because each different neuron is responding to a different frequency. At the cochlear nucleus, you have a further enhancement of all these different contrasts. Then at the superior olivary complex, you have coding of binaural cues, very important if you want to localize sound, lateralize sound, combine sounds coming from both sides. Then you have further things like amplitude modulation, binaural cues. Then you have a higher level, in fact, at the thalamus, which is responsible for picking up the slow contrasts that occur in vowels, syllable differences, also extracting certain features. Also very important, integrating the senses. This is something we all often don't think about. What I see, what I hear, what I touch, what I taste, all of this is coming together somewhere in our brain. And the thalamus is very important for doing that. Then you have the primary auditory cortex, the coding of rapid acoustic events such as consonants. So if you have a problem at the level of the cortex, you might have a child that seems to be hearing but is not able to make out differences in fine structure like consonant differences. Also, if you want to have space, like I want to judge how far is someone, three-dimensional aspects, all that happens at the level of as high as the cortex. So you see the ear is nowhere near it. It's happening at the cortex. And then you have very important part, the Wernicke's area, where you have comprehension of spoken language, obviously very important since we want children to communicate and develop verbal language. Another important thing, the corpus callosum, which is interhemispheric connections. So if you hear something like a key jingling and you want to say what it is, you have to have that sound being processed in the right hemisphere. You have to have that impulse going to the left hemisphere for you to be able to label it verbally. Otherwise, you'll be able to hear it, but you won't be able to say what that sound is. So it's very important that the two sides of your brain are correlating information, are transferring information from one to the other side. So phonological processing, auditory figure ground, that means picking up a signal in the presence of the noise. All of these are things that happen with the corpus callosum. So what's the take-home message? Successful outcomes from cochlear implants are not dependent solely on putting those 22, 16 or 12 electrodes into the cochlea or on the presence of neural responses such as NRT or ESRT. Understanding the complexities of anatomy and neurophysiology of the auditory system gives us an edge in troubleshooting, which is very important for us, especially as audiologists and speech pathologists and any professional who has anything to do with cochlear implants. Thank you.